Today on Aqua Kids,
the Aqua Kids are back on the York River with Dr. Willy Reay of the Virginia Institute of Marine Science, where they learn about the high-tech science behind measuring water quality. Plus, a look at how the Chesapeake Bay's blue crab population is doing.
Aqua Kids, doing what we have to do. Saving our earth, land, air, and sea. All we gotta do is keep it green and blue. For me and you. Aqua Kids.
This is Aqua Kids, and I'm Hannah Jones. Hey guys!
Hey!
So we've got another beautiful day on the York River. What would you like to learn about?
Water quality?
How gliders can be used for aquatic research.
and diseases that affect the blue crab.
I think we can cover all of those. You ready to start the day?
Yeah.
Let's
Let's do
go.
it.
We hopped aboard and headed out to the York River. There, we will meet up with Dr. Willy Reay from the Virginia Institute of Marine Science. We're here with Willy Reay from the Virginia Institute of Marine Science. Hey, Willy.
How you doing?
Great. Selena has some questions about water quality.
Sure.
Yeah, I was wondering how the water's doing on the York River.
Well, it all depends what water quality issue you're addressing here. So the York River is very much like most of the estuaries in the United States. It has a lot of issues, some good, some bad. In the York River, we're primarily interested in a little bit excess nutrients, which causes things such as harmful algal blooms. And another issue that we're having in the York River is the lack of water clarity. Most of the contaminants that we're seeing are things such as too much sediment, too many nutrients bleeding off the land. We also have some issues with toxics on the York River, but that's fairly small. And then another issue are bacteria, such as when you hear beach closures, things like that. Many things going on in the York River, again, like a lot of estuaries in the United States, are dominated by non-point source issues.
What's a non-point source issue?
Okay, I guess the simplest way to explain it is to explain what a point source issue is. A point source issue is I can walk right up to a pipe and show you, and you'll see that contaminant. It's very easy to measure. We can measure how much of that is coming in, and we know the location. A good example would be sewage treatment plants, okay? And you can see a lot of activity has occurred in those areas to clean those up, to reduce those nutrients coming in. Non-point source issue is another beast. It's very difficult to control. And the reason why is it's leaking off the land. It may be coming in from stormwater runoff, groundwater drainage, atmospheric, from the rainfall coming in. So you can see now it's a very different type of contaminant source to deal with, very difficult.
So an easy way to remember it is a non-point source is bigger and harder to identify, and a point source is something that we can go to and actually look at.
That's right. So to give you an example, the non-point issue for the bay would be 6 states are contributing to the Chesapeake Bay in terms of non-point source loadings, okay?
So how do you go about sampling the water quality?
So what we're going to do today is show you a little bit of the progression that technology's enabled us. So it has changed over time.
Great. We'll be right back to check out the latest technology.
Now it's time for Aqua Quiz with your host, Drew Cruz.
I'm your host, Drew Cruz, and now it's time to test your knowledge with Aqua Quiz. Water quality sustains the ecological processes that support native fish populations, vegetation, wetlands, bird life, and our own health. When monitoring water quality, what factor is not considered critical? Is it A, turbidity, B, dissolved oxygen, C, pH, D, temperature, or E, evaporation? Don't float off. I'll have the answer after the break.
Aqua Kids will be right back.
Did you come up with the answer? What factor is not critical when determining water quality? It's E, evaporation. Evaporation is not a critical factor in determining water quality. I'll see you next week with another Aqua Quiz.
Welcome back to Aqua Kids. I'm going to turn it over to Betty, and she's going to explain the latest technology in testing water quality.
Selena and Andrew, I'm going to talk to you today a little bit about technologies in water quality measurement. And one of the simplest that goes back many, many years ago is the Secchi disc.
How does it work?
Well, first of all, you go to the side of the boat. We lower the Secchi disc through the water column until we can't see it anymore. Right now you can see the black and the white. Once it disappears, and you can no longer see the black and the white, which I can't see it right now. You bring it up until you can just barely see it. Can you guys see it?
Yep.
All right. We take a mark, and then this is in grades of 10 centimeters. So our Secchi depth is 10, 20, 30, 40, 50, 60, 70, 80, 90, 1 meter, 10, 20. It's 1.2 meters.
What type of visibility do you normally have this time of year?
Out here in the middle of the river, a meter. It can be better, it can be worse, depending on the wind.
So why did the Secchi disc disappear?
Well, that's because there's a lot of different things in the water column that can block the sunlight. For example, we have a lot of just suspended solids, which are mostly sediments and that sort of thing. And this here shows you how much sediment can be in the water when we filter it onto a filter disc. The other thing that can be on the water, in the water, is chlorophyll. Chlorophyll is a plant pigment which can block the sunlight as well. Another way that we measure light is using this light core sensor. This is a submerged sensor that will measure the light under the water column, and then in conjunction with that, we use a depth sensor. So we lower it into the water column, and first we get a reading close to the surface. You push the enter button, and we've recorded the light reading from the underwater sensor as well as the depth sensor. Then we go to our next depth, which in this case is another 10 centimeters down.
Press enter.
Yep, go ahead.
All right, just did it.
Okay, one more reading and that should be it.
I've got it.
So is this device measuring the intensity of light at the different depths? It seems a bit more advanced than the Secchi disc.
Yes, it is. What it's doing is we take these numbers, the data that we collected, and plug it into an equation back in the lab, and it gives us an attenuation, how light is attenuated through the water column and decreases as you go down in depth.
Why is water clarity important?
Water clarity is very important in order for submerged aquatic vegetation to grow. If there's not enough light, then our plants won't grow. Down here in the mouth of the York River, water quality is good, and we have a lot of seagrasses growing in the shallow areas. But if you head upriver, there's not enough light up there for the plants to grow. Historically, there was.
Before we go to a break, Willy's going to show us one more device that measures water quality.
Okay guys, I just wanted to show you one more of our stations that we're using. It uses the same technology, but the difference is the surface mapper is run about once a month and can let you know what the whole river is doing at that time. These are what we call our continuous stations. They're out there 24 hours a day, year round. And so this information will give us about, if you take a single parameter, let's say salinity, during the day that'll give us 96 measures of salinity. Over the year that's going to be close to about 35,000 measurements. If you add all those sensors together, we're talking over 250,000 water quality measurements a year coming from that one station.
Is the station only here on the York River?
No, well, it is. These are going up the York River. We've got about 6 of these on the York River, but this is part of a national program. The National Estuarine Research Reserve, it has a system-wide monitoring program, and the measurements from those 28 estuaries are going to be tied into a larger effort called the observing system.
Where do they find that data?
Well, what you can do, there's a number of places to find it, but we actually have an app sitting on our NOAA Research Reserve station, so you can actually pick one of our research reserves around the nation and see what's going on right now.
Oh, cool. You guys want to try?
Yeah.
Okay. Oh my goodness, as far away as Alaska. And it's up to date, and it says the water temperature is 9 degrees Celsius and the salinity is 31.4 PSU. That's amazing.
Wow.
Polar bears are the largest land carnivore on Earth and live almost exclusively on sea ice that forms along the Arctic coast in places such as Alaska, Canada, and Russia. With a heavy pelt, thick layer of blubber, and fur on the bottom of their feet, these aggressive predators are designed for the cold. Polar bear prey on a variety of different animals, including beluga whale and walrus. However, seal is their primary food source. Remains left from a polar bear meal sustain a host of Arctic scavengers. There are only about 25,000 polar bears left in the wild. Global warming has stunted the development of sea ice, making it nearly impossible for this animal to hunt and thrive. Sadly, at their current rate of decline, these fantastic creatures could be extinct within 3 decades. Find out ways you can help the polar bear and other apex predators on our website.
Hannah here. For more information on today's show, go to aquakids.tv.
Aqua Kids will be right back. Welcome back to Aqua Kids.
This is Selena. On this week's Eco Defenders, we have Fernando Aguilar, who is the youth manager for the Tree Musketeers. Hi, Fernando.
Hi, Selena.
Can you tell me a little bit about the history of Tree Musketeers?
Well, Tree Musketeers was founded back in 1987 by 13 Brownie Girl Scouts. These 13 Brownie Girl Scouts started this organization to show that you don't have to be an adult to make a difference.
Your program is entirely youth-led. Why do you feel like that's important?
Kids can actually see the opportunity where most adults can't.
What are some of the goals of your program?
Well, some of our goals of our program is public education, healthy earth, youth leadership, and global partnership, where we get to have people all over the world plant trees. It's actually one of our programs called the 3x3 campaign, where we plant 3 million trees by 3 million youth.
How many trees have you guys planted so far?
We're almost there at 1 million. We're really close. What kids can also do is they can start a Partners for the Planet club. Partners for the Planet clubs is a club that you can create in your community. From there you can get how-to kits to how to plant a tree properly, and you get to be in partnership with 1 million planters around the world.
Can you share the most interesting, challenging, or unusual project that you guys have had?
One of the most interesting was we're doing our weekly volunteer events, and one of our volunteers actually found an endangered animal, the legless lizard. Projects like these are really important for kids because it gives them a chance to lead a group, to learn more about the community and what they can do to help it. Especially now with pollution in the news, global warming, and kids need to understand that you don't have to be an adult to change things. You can impact your community, and no matter how old you are.
Thank you, Fernando. That's really good information for our viewers.
You're welcome.
We're at the VINS Laboratory for Ocean Sensing. Let's find out what the Aqua Kids are learning.
Donglai, could you tell us about what we have here?
This is an underwater robot, and it's called an underwater glider that goes up and down the ocean, making measurements of the physical and biological properties of seawater.
So how do you control this?
It's an autonomous vehicle, but you can control this via satellite, issue new commands and new missions.
So since it's autonomous, what inside the glider enables it to go to the surface and go down to the deepest depths?
The glider has a pressure sensor on board, and the pressure sensor tell the glider how deep it is. Once it goes down to a deep depth, the pressure sensor will tell the buoyancy pump on the glider to push the water in a little cup out of the glider, make it light again, and will come up to the surface. And when it reaches the surface, the buoyancy pump will draw the water back into that little cup and make the glider heavy again, and will dive down. So it goes up and down the water until the mission is completed.
So this glider is shaped like a torpedo. Does it go really fast?
You might think so, but the glider does not go very fast at all. It only goes at a speed of half a knot.
Wow.
Half a knot? That's barely moving. Why can't it go faster?
The glider trades speed for efficiency. In the water, it actually gets the equivalent of over 10,000 miles a gallon.
Well, that's amazing mileage. How long can it stay underwater?
The glider can stay underwater anywhere between a few weeks to months at a time.
So while it's deployed underwater, how often is it collecting data?
It collects data, depending on the type of sensor, it collects data on average once a second.
What data does it collect?
It collects data such as water temperature, salinity, and color. Color is affected by the amount of plankton in the water, so based on the fluorescence from the water, we can tell how much plankton there is in the water.
This is a pretty slick looking machine. Where are the sensors?
Great question. The center of the glider is the science bay, and the sensors are located on two sides of the science bay. Here's one that measures temperature and salinity. On the other side is a sensor that measures ocean color.
So before we take a break, does anyone have a last question?
I have one. So where in the world have your gliders traveled?
We have used our gliders to explore the submarine canyons in the Atlantic Ocean and Arctic Ocean.
Thanks for that wonderful information. Don't go away. More with Aqua Kids when we come back.
Hi, Drew here. Join the Aqua Kids next week as we talk about the devastating effects of climate change and sea level rise.
I'm Jalen, and this is Earth Edition. If you had an aquarium and didn't keep the water clean, you wouldn't expect the fish to live, right? That's the same with our world's aquarium. All of the streams, rivers, bays, and oceans that are home to thousands of fish, plants, and mammals. All over the world, scientists are monitoring the quality of the water that supports the plants, fish, and animals who depend on it. There are natural reasons for declining water quality, but sadly, poor water quality is largely our fault. Overbuilding, industry, air pollution, litter and runoff of pesticides, oils, and detergents are all man-made problems. Each of us can do our part to reduce pollution. I'm Jalen, and this has been Earth Edition.
Welcome back to Aqua Kids.
I'm here with field biologist Michael Sebo. So Michael, tell me what's going on here.
Right now, what we have Mandy Bromelo, who's one of our graduate students in the Marine Crustacean Ecology program here at VIMS, and she is doing an experiment right now where she is tethering crabs, and on one portion of these she has some GoPro cameras that are out that are actually trying to figure out what it is that is preying upon some of these crabs that she's placed out in the grass beds here.
All right, so I'm about to hop in with Mandy and we're gonna see what she does. So here we go. Woo! All right, show us what you got here.
All right, so here we have a marker PVC, which is basically how we find our poles once we put them in. And then I have a garden stake right here that has another piece of PVC on it. And this is exactly where we can mount the GoPro. So if we open up the mount, we slip it right on, screw it in, and the GoPro film goes right over where we put the crab in. So we have a little stake with fishing line so we can find it. Stick it right under, crab slides onto this on a swivel. It's tethered there, stick it under, hit go, and then we've got video going for about 5 hours with a 2-second photo time lapse.
Wow, do you use live bait with the crabs?
We don't use any bait, so basically the crabs that we're tethering really are the bait. So we put juvenile crabs out, and then we're taking video to see what's actually eating them in these seagrass beds.
Got it, so you use the actual crab as bait for other crabs?
Basically, yeah, crabs and potentially other fish, but we're trying to figure out actually what is eating them, so that's kind of the purpose of the study.
Drew just returned from his crab survey, and now he's going to learn more about them, including where they live. Crab condos.
Crab condos? Hannah, what are you talking about?
You'll learn from Jeff.
So this is a crab condo. You can see it's basically just a cup with some mesh on it. And what we're doing with these is we're putting juvenile crabs in them and then putting them in our little minnow pots right over here. You can see the minnow pot.
Oh, yeah.
And these will be stacked up in the minnow pot, and then we'll put this in the water. We can use this on our juvenile crabs, and here you can see our juvenile crabs. Our crabs are marked or tagged with little orange or golden tags, and that allows us to understand when they molt or not.
And why are you tagging them?
That is a really good question, Drew. What we're interested in is whether the crabs obtain a disease, whether they get infected with a disease, and whether that happens during molting.
What is this disease?
The disease is a rather complicated, has a rather complicated name. It's called hematodinium. It is a parasitic dinoflagellate, so it's a protist, a protozoan, and it lives in the blood of the crabs and eventually kills them. At some point, the crabs obtain it, and they get it when they're young like this. And we want to know exactly when they get it so that we can study that process.
So do you take blood samples from these guys?
We do, but these little ones are hard to get blood from, so we end up having to process them in a different way. But, Drew, today I brought in some adults for you to try your hand at bleeding. Oh, okay, there's your crab, Drew, so go ahead and do what I did. That's right. Sterilize the surface you're going to take the blood from. And here we're just using a little bit of ethanol. Alcohol.
Nice and docile, I like that.
Yes, it's nice. We've put her on ice to make it so she's easy to handle. and not too far in, and you make sure you keep track of your sight. It's not as easy as it looks, is it?
Right. Oh, I'm getting a little.
All right, excellent. Okay, that's good for now. It's hard to get a good sample when you're just, you got some. And then just go ahead and put a few drops on that slide right next to the,
This one right here?
Yep, right there.
Hey, Drew, how's it going?
Hannah, don't you have things to do?
So, Drew, that blood smear, let's take a look at it under the microscope here. So just go ahead and put it on the stage. So what you're seeing there, Drew, is the parasite. It's the one that lives in the blood, like I was telling you, in the crab. And it can rapidly grow in the crab and eventually will kill it.
Is it contagious?
It is contagious, but we're still trying to figure out how it's transmitted, and that's why we're using the little juvenile crabs in the condos to see if we can get them to become infected in the field.
Well, Jeff, thank you so much. You're doing great work to help save the crab population.
Thank you, Drew. I enjoyed your visit.
We had a very busy but informative day on the York River. So I hope you guys learned all about the topics you were interested in. How about you, Selena?
I learned that visibility is really important when it comes to water quality.
I learned that gliders can monitor water quality autonomously throughout the world.
I learned that scientists are undergoing studies to determine the parasite that's affecting blue crabs.
Great job, you guys. Remember, it's up to you to keep it green and blue. Help protect our planet, and we'll see you next time on Aqua Kids. Bye!
Aqua Kids, Aqua Kids, doing what we have to do. Saving our earth, land, air, and sea. All we gotta do is keep it green and blue.